Evening and welcome once again. Today is our 1745th day of continuous webinar. Thank you all for joining with us. Today's our speaker is Dr. MASHHOOD. He is BHMS and Diploma in Acupuncture. He is a retired medical officer of Ayush Homeopathy Department, Kerala. And he has taken many classes for us before in Malayalam session also. He's the author of two books. One is Unveiled Micro Secrets and one is The Elements of Human Body. And he is continuously doing research about homeopathic medicine on his own. That is his passion. Actually, that is the passion of Dr. MASHHOOD. I'm inviting Dr. MASHHOOD to international on behalf of International Forum for Promoting Homeopathy. Dr. MASHHOOD, you are welcome. Please. You are audible, sir.
All coordinators, all prayers to our Master Christian Frederick Samuel Hanuman and my teachers, especially Dr. Abdul Latif Sir, who made a foundation for these agro studies in homeopathy. All of you, today I am going to present a very new one. I think still far nobody showed this type of commitment to homeopathy with the plants. Today, I am presenting here some experiments done by me and observations got from the experiments. Clearly, I am showing now here some pictures, especially some crystals. And these pictures says everything than what I am saying. Every picture is an experiment. Later on, I think that completely one by one, I can show something, I can say something to you. Now these pictures say something. First one, introduction. See, please, doctor.
Wait. Yeah, I'll share. Actually, I have to enable the this one. That's why. Wait. Okay. No other co-host, right?
Every plant.
Wait, doctor. I'll make. Okay, you can continue, Dr. Ram Parvesha. Sir, please admit the participants who is coming in. Okay. I made you as a co-host. And anyone else is here? Okay, now I'll share the slide. Can you see my screen?
Yes.
Doctor, it's visible, right, doctor?
Okay, visible.
Okay.
See,
okay.
Crystallized substances present in some plants, mother tinctures, and some changes observed in the crystals of plants after the usage of homeopathic medicine. This is my introduction. Crystallized substances. First, you all may see some crystals of salts in our daily usage. This type of crystals, very minute and microscopic way, are present in small amounts in all leaves and fruits, everything. I studied several plants, and from the introduction, said that my books about these two books are available, and they are all because of these crystals, which got from the medicinal substances. Every plant do not treat every elements in the same manner, but the crystal, how they are situating, how they are present in the plants is not understood. I think these crystals are settled after some solutions prepared, or even in the plants itself, some crystals present. It is a doubt. But in some plants, absorbed directly from the plant juices, the plant parts, some crystals, some plants, only the juices or the solutions got these pictures. My intention, this next slide. The action of homeopathic potencies in plants like chili, peperomia, tapioca, pea, and some other small plants like this Peperomia pellucida. That is the plant species which I selected as a specimen, and it is the mostly used plant in my experiments. That is Peperomia pellucida. The Malayalam is very small pacha, small plant which is used in our natives. It is not as a medicine. It is used sometimes some places as this vegetable. Now, what is the crystallized substances from the mother tinctures? I observed some more, about 60 mother tinctures of our homeopathic potencies, which is got from the pharmacy, and the crystals are unique and they are identical for that particular mother tincture. That was very interesting. That also I'm intending to show in later classes. Here the most important part of this study is the changes occurred, changes observed. I saw from the experimental plants after using very high dilutions, very high potentialized medicines. That is the importance of this study. See the next, next slide. Here, no, before that.
Second one, what you are telling, doctor? Second slide we already shared, no?
Yes.
This one third.
Yes. This is one. What are the methods? What are the tests done with these experiments? Is very short. I have done it for a long time, about 13 years, and after that these results are obtained. Till now, I am doing these experiments, and about I think it is 23 years. Last month, I got some pictures also included in this class. It is started from the studies from 2002. That is 2002. I saw Dr. Abdul Latif sir and got some medicines from him, and I started my experiments with some homeopathic medicines, my own. That is especially the tapioca and some other chili plants are used for these experiments. Medicines, here you can see the tapioca stem. Stem that I marked this tapioca stem after 6 months old tapioca. About 64 plants are I planted, and the tapioca plant I gave it some nail marks, because some nail marks, and so after medication, what is the nail marks growing or it is deteriorating? What is the change? From these 15 plants, I gave the tapioca, Silesia vernum, and other chili plants are also studied. This Calcarea carb, Silesia, Calimur. I am directly going to the medicines. These potencies are used for this medicine, this experiment. One is chemical fertilizer and phosphate. This another study, that is how the medicines are acting over fertilizers. Is there any benefits than these fertilizers and growth the changes or something else with these homeopathic medicines? That was my another study. That is for that I used this phosphate fertilizer. that phosphates are especially it is the complementary of silicea. That is why it's selected is phosphate. Also, it is a cheap and we'll get from the fertilizer shop. That is phosphate. Then second is the medicine. I selected the plants and they used for the chilies, one for chemical fertilizer, second medicine and fertilizer used together, then medicine only, then Tonum silicea medicated number 10 pellets, 15 to 30 in number, with 1 ounce water and successed 100, and used around the roots and sprayed on leaves, and 2 weeks later repeated twice. This was my method of experiment. This 1 potency number 40 pills, 15 to 30 in ml number, that is number 10 size pellets, very small medicated pellets, 15 to 30 in number, successed with 1 ounce of water, and it is sprayed and around one that is a 30 centimeter around this, and also to the tip of the stem, that is at the base of the stem also, and sprayed some leaves also. These the growth of the leaves, roots, shedding, everything observed about 4.5 years for chilies and 2 years for this tapioca. That was my experiment, first done. After that, then next slide. Again, now as a procedure, I am saying the results of what the results of obtained. This medicine and fertilizer used plants showed very great extent, good results. Then silicea used plants also showed long life and healthy green color. These 3 points are very important according for the silicea. That is, green leaves preserved, more green showed, and also the lifespan was very elaborated, chili plants. And then the nail marks, which is based on the around the plant, that is, I will show the picture of the nail marks for 15 plants. That is one around the stem. And after 6 months, again studied the growth of the nail mark, a mark which is around the stem. And what is the result? Because I have so much suspicious about this silicea. That is, what is the excess, or it is a similar, it is excess or deficiency. What is the basic of our homeopathic potencies? That is another intention. That's why I used these nail marks after studying, and it was also good results, very great good results. This the characteristic, the substances, the crystallized substances visualized under the microscope are another study, which is from these experimental plants. I collected the leaves after 2 weeks before applying the medicine, and after applying 2 weeks, the leaves are separately placed, crushed, and it is mixed with, as our homeopathic medicine preparation, very strong dilution spirit, and kept it for 8 days. After that, the solution is decanted and kept it for 2 days for separating these crystals. 2 or 3 days it's needed. And after that, these solution, from this solution, 1 or 2 solution drops of the solution is placed on a slide and kept it for drying for about 8 days, minimum 8 days for some crystals. But some crystals, it takes at least 16 days without any moisturizing, which is without any the air damage or from dryness, powder, this dust, and some insects, everything you must keep. And after that, these slides were dried and viewed under the microscope. Especially, we can see these crystals which I showed is usually is very easy to see by under 10 magnification. But some crystals very light and very small, under microscope 10 magnification, 100 magnification, 45 magnification, even then it will be very 1 or 2 millimeter in size. I magnified it with my mobile, and picturized this with the magnificent pictures. That's why it is very clear and vivid. Some crystals observed from the plants with homeopathic medicine used, and then these plants are made into dry and become ash, and from the ash also these crystals revived. These crystals can be seen. That is very important in some cases. So this study, I am not finishing. It is not an end of the study. It is each and every step is to be revived for newer studies. I am still looking for further experiments and things. Here, the absorption of these metals and elements from the plant, from the root side, how it is occurs, along with heavy metals and heavy dosage of these substances, this minute amount of our homeopathic medicines, how it influences on the roots, and how it is helps the plants to reabsorb from the nutrients from the plant. Is it it gives any special differences? Is my study. Then next one, please show the slide, doctor. Slide. This is one with copyright about the copyright about thing. So yes.
You want next one?
Next one. Next.
Okay.
So many parts of this class is presented in a nice conclave 18, 219 on the Kanagakumma Palace, Trivandrum, Kerala. And many of the pictures are the same one, which is published there in 19. But now, even in 25, even now, I am looking, I am getting this sort of crystals even now. That's why I am reviving this class to you all. Because next, this crystal, last class also I showed, nobody may be seen, it's very important crystal of sugar of milk, our sugar of milk. This sugar of milk crystal got it from the first sight of the test slide from my microscope. And later on, several times got this type of crystal. It's a very interesting sugar of milk crystal, our sugar of milk. Next, I am not going very fast. I think that it is better. It is enough for about
This is right. This is the one, right? 7th slide.
Next one. This is also sugar of milk. See the crystal of sugar of milk. What is sugar of milk? To be used. What is it is using this molecular formula about everything? Which later classes I have told you in the Malayalam class. I think all of you know. Nobody is don't know. This sugar of milk is a very important factor of our homeopathic the dispensing, and also its piercing power is most interesting. Like the solubility power of the alcohol, this sugar milk is also a very great friend of us, and that is the sugar of milk. This is another one which I want to show this picture because you have got an idea from what I have seen for these 23 years, and I think that from books and from my classes, these type of crystals are coming further. So it is belladonna. It is a picture from belladonna. This can be seen from our lab. From once I have a chance to show our director in presence of director. She was there at that time DMO. And now she is not the director. I was last director. And this time I could a very imminent presentation of these crystals. I took a medicine from belladonna from the lab of the hospital, and a function was there at the time with this hospital. And I took a belladonna tincture, and at once I showed these crystals. This is belladonna. The next from the crystals usually seen in the belladonna crystal. This is from pharmacy. We got this. It is from the tincture. Next, please, doctor. Next.
This one, right?
Yes, it is calendula. This plant is, this is got from the picture is the typical crystal of calendula tincture. This is a plant, and this is the medicine we know, calendula. Next, calendula. See, this is an opaque, this is not a transparent crystal like some other crystals which I will show. This is an opaque crystal, white powder like a crystal, very soluble. These crystals are soluble in water. Many of them are can be picked with a very minute needles. I put to change their places. From their original place, it will be moved from that place. I can, and it is soluble. And again, regaining this state after sometimes if it is dry with a untouchable place. Okay, next. This is calendula. Next. Okay, next. This is a very wonderful crystal from Artica urans. See the very typical crystal. Everyone sees that, say, or look, crystal means certain types of square-like or cube-like thing which is transparent and it is soluble, and it has its own layer is called lattice. This lattice formation is very important. But now see the crystal, it is very much important. That is Artica urans. Our medicine Artica urans crystals. Next, this is tremendous crystals are seen from one drop of this Artica urans. This is a very wonderful crystal of this euphrasia. A very old solution was there in my lab, in my dispensary, when I went to this Idiki district for that class for my job. At that time, on 2011, I got this crystal that is euphrasia. It's a very important crystal from 2011. I got this. Okay, next. Several crystals are there, several types of crystals. This is one type of crystal which I told you. These are only examples, very showing. And next, I have classes, for the classes, I will show different types of crystals which we can, if time allows. Okay, next, doctor. Next picture. This is a thing which I want to emphasize it very more because in my conclave also this presentation, I presented this picture. You can see this as like an animal. In his stomach, there is a very small crystal. Can you see? I can note. I don't know. All of you are visible. This small crystal. Next slide, please, doctor. It is enlarged in next slide. See, a small blade-like structure.
Yes, slides are visible.
Yes, this slide is a very important crystal. I was so much astonished what it is because I used a very small amount of sugar of milk. Very small, you know how I am testing this homeopathically to see the homeopathicity, how I am doing these succussions. I did succussion even for 100 succussions. At that time, every experiment I will do a succussion. Very minute, very pinpoint sugar of milk with a 3/4 of the ounce of water and succussed 100. I took these medicines with the water boiled and cooled, very pure water. And the succussed and this sugar of milk is applied both for a chili plant. That chili, around the chili and the leaves not at all sprayed because in these experiments, the leaves are kept very clearly without insects and some other dust because doing the microscopic examination, I am only just washing the and drying and made it into the solution. So microscopic study even is this so so much care is taken given to these experiments. Even then, this crystal was an astonishing one of sugar of milk. That first time that I showed the crystal of the sugar of milk, we are all seen. That is similar to almost similar. I think that this is the leaf, not the fruits. Some fruits may show because of the sugar contents, ripened fruits. I observe some things, but it is not the leaf. It is leaf. Next, this is another picture of before, you can, I think that there is a word before. That is, some pictures I am showing before doing any medicine. That is fresh plants. Next, this is a picture of leaf which is taken from a chili plant, and it is grinded and mixed with highly potent, highly strong spirit. And after that, this solution, the liquid is kept with 2 days and 16 days for the drying. And after that, visualized this picture. This is, see, there is very small amount of crystals there. There are some salts or some other things of the chili plant leaf. Next, this is also same like that is, you can see some red small things were there. It is also a very leaf. This is also a leaf of the thing. Next, before, next slide, Doc. This you can also see the before one. See before this one. This before one, doctor. Okay. Before this picture, please. One. Yeah. See the small one, which is this, if it is enlarged, you can magnify it. You can see a very green some flame like crystals, 3 or 4 in number, which are crossed like this is a usual crystal of chili plants. It's a usual sign of chili plants, leaf from the leaves. Next, the next picture is an important one. Same, some materials is there. These materials are also usually seen some chili plants. These even every crystal is not a single solution or single slide. It is got from several size and several plant solutions. I was in the habit of doing same experiment for several experiments at a time. Silesia, some other medicines are in a group, some another thing for chilies, some another group. Then this peperomia plants, another set of at which I was while doing, I don't want know what to do. For these 13 years, I was completely absorbed by these studies. And very little time was I saw Dr. Latif sir, but again, our Dr. Leela Vadi, she was our great principal of from college, Homeopathy college, and every day I think that if I get a new one, I will go and see her and say everything. This she was so much helping, and she told me everything to record and record. That was why I recorded in a very elaborated manner there. It is here, and which helped me for the presentation in the Irish Conclave. The first I was rejected from the Irish Conclave, but again there was a flood in 18. That after that flood, I was so much weak. After that, I was in Idiki district in a very hard situation. At that time, flood. I think that after 2 or 3 days, the food also will be not good. We will be dying with food and water, I think. That situation came in 18. Even then, we restored from this. And at that time also, even in my indoors, I was doing these experiments. This is a very important one. The next. This side is again after. That is the medicine given. And after that same chili plants, same pill chili. The first picture, you see, that same picture, same plant, which is after this medicine. This is a very small crystals, white type crystals in this right side of the angle, triangular white crystals, which is amorphous. That means it is soluble, but not crystalline, though not transparent, but it is soluble in water, and it is seen, these crystals. Then next, it is also a common, it is an uncommon one. That is what I want to say. This is due to Silesia applied plant, that is 1 potency, that is 4 pellets, so 10 number 10 pellets, size pellets 15 to 30. Usually worked at least 20 pellets in 1 ounce of water and successed, and the chili plants around it is used. The next slide is very important. This is also an unusual thing which is sought. every day and everybody, we may say that the homeopathic medicines may dilute or dissolve or destroy the extraordinary chemicals and metals from the plant. But in my experiments, several experiments, then it will be a nutrient or natural nurturing thing for the plant. That may be the thing these crystals are more. Even in this plant, this type of rod-like one is a very rare in chili plants. Even in fruits or leaves or roots, shoots, everything, nothing. I studied these even in roots also. There is nothing like these of this before or without using any indulgence. Indulgence, that I want to say that because if you are using some urea or some other things, some phosphate fertilizer, some other things which is in a magnesium sulfate, etc., on the root in a very careful manner, then observed there are also sometimes this type of reserved particles may be there. Sometimes these particles may be reserved particles or the materials or of the metabolism. Sometimes it may be for the protective action. What is it? I don't know. Several times, which is very easy to understand that these are the metabolic products. Every day, anyway, it is a metabolic product. That is the change occurs due to these medicines. Is it a true one, 90% or 100%? I don't know. Even now, I am saying that these are typically of the because of these high dilution medicines. Even in one thing I can say that is Silesia is a very wonderful remedy which I studied very long time. And if you can see the next plan, next one. This is the picture which next one. This is the picture which I want to say that you can always do. Nobody will even now these things I think. But next day onwards, every children or students very much enthusiastic can do this experiment. See the first one, the color is very yellow. That is no green. The second one is so much green. Why? It is because I am saying that 200 solution sometimes, but 1 potency of Silesia will definitely show this type of green appearance. Then Dr. I am hiding these for these 23 years. And now I am showing these. Why? I don't know. This picture I took from the last time, that is 221 or 22, after returning from Idiki. This was first done in my Cornish conclave. I explained in this literature. I told you that the green color is preserved as such. The green color is very much noticed in this, and then this experiment with it is a very that is the manner that is number 1 sugar of milk, number 1 globules, number 2 globules, number 3 only water control, and then water spray, then 4 Silesia. These experiments done elaborately for several times because I was so much interested because of this green color. Everybody can say that is Silesia one. If it is Silesia, it will do this type of green. If sugar of milk does not show, it will becomes yellow color. That Dr. Abdul are several times told me this papaya, this bitter cucumber leaves. It was the specimen for him to his experiments in the first time, and he saw so many times this yellow color is mentioned, and it was I think that it was his first sight from his experiment. I saw this, but he doesn't know this Silesia he will do. I told him to about this Silesia one time about the chili plants, and this I didn't even now before his retrieval. I have an opportunity to show a crystal with the meserium and some other things, and also his plant medicines. Also, I tried and gave him. I sent him some pictures before his 2 or 3 days. Okay, next, this experiment is a very important one. There will not get any green color with globules, water or sugar milk. You will get only with the Silesia this type of green difference. Okay, and, only spray, even at least spray minimum. That sugar coated. That means medicated pills are used now here. That is an important one. But the sugar globules also same like used will not show any color because now after the globules it showed yellow color. But after silesia it becomes again very great this green preserved. Again, another thing which generally I want to say that if we use a sugar of milk or a placebo first, then we use the medicine, it will not affect, it will not show any reaction. That is many time I observe. But if you try silesia, then or any medicine, then you apply saccharum lactis or sugar globules, placebo, it will again react. What is that? That is the effect of placebo effect also can be proved by these experiments. Next, this is a combination of my combination. I made a formulation and it is kept in with me for a long time and I am trying again now. That is the first one is it's a diseased plant. This is 14 or 15 days after. See the difference. Very growth. These old leaves are here and there. You can see, and the difference is this 20, 22, 23 days. It becomes this type of difference. See the cotyledon leaf. That means the first leaf is still preserved. That is the importance of this medicine. Cotyledon leaf means the first leaf. That is also even preserved from these experiments. See the next. This plant is was a long-lasting living one and gave many fruits. Next, I am starting this picture. That is sorry, I got the introduction. This is a picture which for your reference, I can show this. Doctor, next one. Sorry. Before first one. Sorry, that picture you have shown that this is a natural normal peperomia plant. This normal peperomia plant is a plant which you can see this plant. Peperomia means Peperomia pellucida is the species which I selected, and these leaves are this experiment in a very short time. I will say that these plants selected in a routine manner of similar size from similar place, and these peperomia plants are washed very clean with the roots without damage, and we placed in the 1 ounce water bottle with heated water, boiled water, and the drops, if it is using drops, then drops will be mixed and sucked 100, and the plant is placed there for 2 weeks or 3 weeks until the plant dies or it may grow. Next, again, the medicine, if it is globules, then 4 or 5, number 10, 4 pellets, number 40, or 10 into 12 or 8 pellets. It is very reduced because the plant is so poor. The roots are immersed in it, and the shoot, root, and the leaves changes are observed. This is a very, very vast study, and I will definitely show you all these experiments in a separate manner. This is a normal crystals of the Peperomia pellucida. You see, this may be get from some other places also. Sometimes you can see these crystals of this Peperomia microscopic crystals. This is the actually from anywhere. If we get a plant and do this experiment with a microscope, you will see this is the natural crystals. It is seen very fresh plants without any medicine. This is, you remember, please see this picture, you remember in mind. Later on, we are seeing some other experiments, the plants. See the next, doctor. Next, please, doctor. I think that one picture is missed. Before this
one, globule, then water.
Oh no, before which one? Before it.
This one we already saw, no? This one. This is peperonia and globule, then water.
No, then before one. Before that, that's meserium. Before that. before globule.
This one?
Yes, yes. This is a very wonderful picture of Mesurium vannam. See the picture, very important one. That Mesurium vannam, it is drops. I used vannam drops. It's from Schwab pharmacy. Very late experiment. 2022. It is also used in my mix, I think. You all see later. I will show some classes, or if it is time allotted. Please say this is a very important, wonderful crystal of separation. It is a crystal from Mesurium vannam, from peperomia plant leaves. There is no typical change of leaves, shoots, or roots without any decay, but the leaves as such, it is preserved for a long time. And also this Mesurium, this plant showed this type of crystal. This is a very special crystal. Even now, it is not repeated. No other plant showed this type of crystal. This is a separate crystal. So Mesurium vannam. Next, Dr. Fresh. No. The globule. Yes, this is the picture. One is that is globule. Globule means natural globule, normal only globules, no medicine. This is a picture I got from the peperomia plant. This is also a peperomia leaf. Next, very speedily we can to one. This is water control. There is no medicine, only placed in pure water. There is also very slight changes and very normal small crystals may be sometimes see very small, but it is organic matter. I think nothing is seen. Next, water. Next, this is fresh from land. It is not from water. It is land. See, the first slide I told you, please remember. Likewise, these picture also some organic matter and very small small crystals are present from land. Fresh peperomia plant land. No medicine. Next. Next, please, doctor. This is certain other type of experiment which I am done. It is because of the because this no, the next one is fresh land, and next one is spirit. Is it spirit? This picture is it is not the next before one.
This is mineral bio. Which one you want? This one.
No, next before that mineral.
Spirit.
Spirit. This is also as a special study with used only spirit, only the medicine placebo, one drop. Sometimes I am using the spirit with the medicated globules spirit. That is usually I am doing the control because our medicines are like this spirit medicated. So medicines even medicine, is it medicine or not? How can we say? So that is another experiment that is with a spirit and a neutral spirit. These is very small type of crystals are established. Well, how I don't know. Spirit shows very small light difference. Spirit. Next. This picture is an experiment done especially for very late. That is from cement sand. You know, for the concrete work we are using that a sort of a new type of cement sand. That sand is filled in the bottle and also the plant is placed. And before the leaves are collected and studied, after several days, this plant is grown and water is poured and the sand material, how the sand material is absorbed through the medicine and it is, we see the minerals from the sand salt can be seen. Sometimes maybe calcium silicate or some silica substances it is seen. This, for example, for your vision, I show this plant. Next one, sir, doctor. This is I have shown in the Malayalam classes. This is from sugar of milk. A plant is placed in the very minute amount of this sugar of milk, and the plant is studied with a microscopically. These juices and the plant's solution typically shown this crystal. Again, next this again I am saying that please remember the central one, that is a tablet-like one is there. Please remember, I will say something about this later. See, this is sugar of milk only. Next. This, I want to say this, even now I am doesn't told to anybody, but now this is a crystal See the color, and it is from Kalimur. This is from Kalimur 6X, and several plants, and every plant. This is my, I thought that it will be a new one to present in some other journal. But in my books, about 56 plants, I observed this thing. And last time, I showed some professors, and they are not, they gave me a chance, my book in this Karivatam University Library and the University Library, Public Library, these three places and proposed bookstall everywhere. These libraries, they promoted to this book, because this experiment is also, even now I am doing, to all leaves. This is Kalimur. It gives a separate entity to my experiment because of this crystal, this color. That I will say if anybody interested later. Please next, doctor. Please next. Next is, it is a fresh in sun. Even though it is fresh in sun, see the crystals very elaborately seen because of calcarea carb. That is carbon powder. Calcarea carb powder, kakkapodi, Malayalam. That is from the fertilizer shop we can get for reducing the alkalinity of the plant. From reducing the the acidity and promoting alkalinity, we are using this substance. So I used the very small spoon to a group of peperomia plant in a bucket and collected that plant. It showed very typical calcarea. It is calcareous. It is calcium. Calcium in a abundant manner, in a bunch of, in an elaborated manner, these crystals are settled because of this is opaque and white. That is soluble. Calcarea carb. This is fresh in sun. Very great sun. It is in a very crisis, I think. So it is taking these crystals for their AC or for communication. These are all doubts. They can say so many doubts are there. I am still in confusion whether these plants are communicating with these crystals, or they are preserving their food, or for doing some experiments, studies which I read from a journal, that plants communicate their communication with these experiments, with these minerals by roots and around the soil, not by the leaves. I saw these things from the leaves, from the roots around these things are. If Mr. Dr. Latif Sir, Abdul Latif Sir is here, I am so eager to say him too a very important thing about this communication of these plants, because his root, through the roots he applied some medicines, and again I used his substance in for a separate study, and I understood a very important one. That may be a positive side thing sometimes, but it is hiding now. Next time, if time allows, I can. This is also for communication or for observation or as a food material or metabolic. The next. This is another crystal I have shown last Malayalam class. This is very interesting. That is 50M Silicea in Peperomia delucida plant. See the 14 or 40 days after, Silicea plants will be there for several days in bottle Peperomia. This Peperomia plant will not have any change or any thing. That will not, the roots will be there. In my studies, Silicea 1 potency and 200 is everlasting one. At least a tip of the growing tip of the leaf may be preserved very green for about 6 months. All the sometimes with the fungal infection or some other things. Fungal infection is the most important side target of this Silicea. A separate type of fungus which is against the Silicea was also reported from the other parts of science I have heard. This was known to Dr. Sir also. And Dr. Ramaswamy, I think our great scientist of the rice and paddy field from Tamil Nadu, was Indian scientist, first scientist of agriculture. He wrote a article in Hindu when I was in Idiki. I don't know how Anyway, I got that paper and I have an opportunity to read that. He told that about the silica. When I was taking classes in medical college, homeopathy medical college, Dr. Divan Haricha was wrote an article in a medical pharmacy, medical one in our Trivandrum Homeopathy Medical College library. That also accidentally I got that. That also he says the wonders of the silica, Silicea. Very wonderful picture he wrote about this Silicea. Even now it is not. I think not. It is not all produced, placed in the field of homeopathy, agro care or agro studies. This is 50 potency Silicea. Again, this is the elaborated one. Same picture, but another slide. See, the small one also is there. This is a very wonderful Silicea. It is a typical crystal. It is also from Velucida. See the small, very small square-like pictures. But now it is in very important one. That is a very long one. Next, doctor. Next. This is Amaranthus. No, every day. No, this is Amaranthus. Next slide. This is before using medicine. See the round, very fleshy leaves. Next one. This is elongated. See, Silicea applied plant shows this typical elongation. There is another. It is a very characteristic one. That is elongation. Elongation. This shoot also in a very climbing plants and some other plants. It is typically you can see this Silicea vannam. It is elongation of the same plant group. This is Amaranthus. Next. This is a plant which is diseased and I got it is from Idiki in Adimali. You know, the place Adimali from this is vegetable shop. There is this plant, this along this all this setup I got it is this for this 80. I think that he take me 80 rupees. See, it's very interesting because blue plants are my experimental specific site, specific target because blue will definitely give natural changes, but our medicines will give very confined, very definite that is best critical changes. That is why I selected this plant for this. And again, next, see that plant was very well withered, and after that, all leaves left and all fruits left. Again, this is revived with this medicine, Silicea vannam only. This plant. See, the same plant. Next. See, I told you from the Malayalam class that the Kalimur plant, which is revived after for a long time, it was there, and it was withered for a long time, and then half of the time, half of the plant becomes the natural. It is a blue plant. It is the plant mother plant was blue. The fruits are blue and become red. Now see the yellow color of this apple. I called this plant as apple chili. This denoted. This is the child of that plant. Last class I told you about the Kalimur and I revived it with the Silicea 50M, calcarea carb, sorry, Silicea vannam, calcarea carb 10M, about some other fertilizers, biofertilizers, everything. I revived and also the rain is the main factor which revived that plant and sun, and it is the daughter of that plant. Again, next generation also these yellow color retain. There is no red color. What is it? This is what I want to say. No medicine, no other fertilizers, no sprays. All plants, all fruits becomes this yellow. No one is become red. Again, another thing is this type of fruits astonished me, and I told you I gave and Dr. Leelavati madam, and these plants showed only a small part of lesion on the side, and it never withered or become fallen off. It is decaying with that plant itself, not drying easily. So much spore, very good edible chili also. See the next doctor. I want to complete this class itself. That's why I am going speedily. This is a reviving one. It is going. See the VIBG or VIBG. That is violet, indigo, blue, green, orange, red. Last one is red, but it is little bit one left one, that is yellow, not orange. This orange color is also written. See the color of the plant blue. It is the plant of the mother plant, that apple plant chili. Then apple chili revived, and it is to another one. This becomes this type of plant. Then last time before this picture, it was apple itself. The shape is retained, but now the shape is gone. To this, and the position is also important. See, don't I am studying these things. The color of the leaves, the position is upward. See, not downward. This is influenced by, especially I want to say, Calimur. Calimur 6X. No biochemics other. No sugar of milk. This is a tablet. You see the tablet 1 grain or 3 grain tablet is 99.9% is sugar of milk. Not an amount of this Calimur is present in there, 6X even. But how this occurred? It is true, and I was repeatedly seeing this one from the 3rd generation. It's the 2nd generation. So, next, doctor. See the above plant, the 3rd before again before one. That is, it is the blue plant. This side, see the upward direction. This is the 2nd generation, upward direction, yellow color only, not downward. See, the plant is round. This is another plant, not apple chili, round blue. Now, it is also typically seen. It is from, not from Idiki. It is, these plants are 1 and 2 and 3 plants, chili plants, where I was in 204 when I was teaching in Homeopathy Medical College. For 2 or 3 years, I think only 2 years, I got 2.5 years. That time I am doing these experiments also. Nobody is not showing. I showed this to the Dr. Isaac, Jose Isaac, I think, in my medical college when I was trying, when I was studying. So, sorry, teaching. But this color difference also at time, somebody also again told the color changes are noticed in flowers, but not in this chili plants. See the color.
Doctor, I'm so sorry. We have only 1 minute more. I think up to 40th slide we'll stop because we have only 1 minute. Otherwise, we'll stop from here and we'll continue afterwards. We have I think 100 slides, right? So up to 40. After 40, we'll continue afterwards. Okay.
Slides are there. Okay. Next time we can continue. Today I am closing this with the happy. Okay, I'm to stop this in this time. Next time, if we get
Thank
a chance.
You. Thank you so much, Dr. Mashood. You have already taken these classes in the Malayalam session, and I also heard your classes. This is wonderful that, as you told, that we are all not doing such experiments. It's new to us. Anyway, thank you so much. And now come again and continue the session with the other slides. Now we are moving to the Malayalam session. Thank you so much on behalf of it, and thank you so much all the participants for joining with us. We'll see you tomorrow, same time. And now we are moving to Malayalam session. I'm inviting Dr. P. Shaila for moderating Malayalam session. And shall I make you as a host, Doctor?
Okay.
Okay, you are the host.